All right, I just want to make sure. I'm, looking forward to the day when I can see each of you in person. I will say it's a lot easier to talk and be more interactive. I guess in some ways you may be lucky because I tend to pick on people when I lecture in terms of having them read the EEGs, but it's harder to do over Zoom. All right, so during the next, hopefully 40 minutes or so, we're going to explore neurochemistry and neurotransmitters. And in particular, I'm going to focus on acetylcholine, dopamine, norepinephrine, serotonin, glutamate, and GABA. My PhD is actually in neurophysiology and neuropharmacology. So this is an area of interest to me. We'll start with a question. Neuropeptide neurotransmitters are synthesized in the neuron cell body, the proximal dendrite, distal dendrite, presynaptic terminal or synapse, and the answer is neuron cell body, and we'll see why. So before we get into this talk, we really need to look at kind of the traditional definitions of neurotransmitters, which basically define them as compounds which must be synthesized and released presynaptically. That makes sense. They must mimic the action of the endogenous compound released on nerve stimulation. And this is traditional definition that relates back to when we were doing these physiological experiments on isolated tissue. And where possible, a pharmacological identity is required where drugs that either potentiate or block postsynaptic responses to the endogenously released agent also act identically to the suspected neurotransmitter. And this is kind of how we pharmacologically isolated and determined what these neurotransmitters were. There are basically two major types of neurotransmitters, which I'm going to talk about. Small molecule neurotransmitters, which are made at the terminals and stored in vesicles. These include acetylcholine, amines, glutamate, and GABA, and larger peptides, which are made in the soma, in the neuron cell body, and then transported in vesicles to the nerve endings. A classic example of this would be calcitonin gene-related peptide, which is about 37 amino acids, and endorphins. A third type of neurotransmitter, which is actually not stored but made on demand, is nitric oxide, and I'm not going to talk about that one. In order to understand these neurotransmitters, we also need to understand the ion channels and ligand-gated channels that they may act upon. Neurotransmitters and drugs typically act on ligand-gated receptors or chemically gated receptors in which the opening and closing is affected by receptors on the external plasma membrane. These are distinguished from leakage channels. These are non-gated passive ion channels, which are continuously open. Remember, potassium is selectively permeable. Voltage-gated channels, where channel opening and closing is affected by the membrane potential and not a ligand. And ion-gated channels, where opening and closing is affected by shifts in intracellular ion concentrations. Let's look at the life cycle of the neurotransmitter. So this would be the life cycle of a small molecule neurotransmitter, which is being produced in the terminal bouton. A neurotransmitter is synthesized. It can be synthesized in the cell body, but are transported downward, as we'll see. But in this case, we'll focus on the small ones, which are produced in the cytoplasm of the terminal bouton. The neurotransmitter is then packaged into a vesicle. This vesicle is released when it fuses with the presynaptic plasma membrane here. This is usually a calcium-mediated process. The neurotransmitter is then released in the synaptic cleft where it works on the postsynaptic receptors. The neurotransmitter then diffuses away or is metabolized and or transported back into the terminus where it can be reused in the production of additional neurotransmitter. The cellular machinery of the neuron suggests that it functions as a secretory cell. Neuronal cytoplasm in the perikaryon and dendrites, but not in the axons, is rich in rough and smooth endoplasmic reticula, further connotating or emphasizing its role as a secretory cell. As I said, these small molecules are synthesized in the terminus, but the enzymes which create them are synthesized in the cell body. They are then transported down the axon via slow axonal transport, and upon reaching the terminus, play a role in the synthesis and packaging of the neurotransmitter prior to release. Peptide transmitters are produced in the cytoplasm of the cell body, and in the endoplasmic reticulum and Golgi apparatus. They're then packaged in vesicles, and those are what get transported. These enzymes and pre-peptide precursors get transported to the terminus, where the enzymes can then modify these pre-peptides to produce the peptide neurotransmitter. These are seldom taken up or reused, and are often degraded by proteolytic enzymes in the synaptic cleft. I just provided you with the figure legend to that figure so that you can, on your own, review that information. All right, some basic concepts, starting with definitions. An agonist is a drug that facilitates the effects of a particular neurotransmitter on the postsynaptic cell. An antagonist is a drug that opposes or inhibits the effects of a particular neurotransmitter on the postsynaptic cell. And drugs can affect neuronal functioning by means other than direct interaction with the synaptic receptor. So to be an agonist, you don't just have to activate that receptor. There are other ways that a drug can serve as an agonist, and we'll explore those, or an antagonist. So drugs that affect the amount of neurotransmitters stored in vesicles can either have agonistic or antagonistic effects. So if a drug facilitates storage, maybe it acts as a precursor, then it can have an agonistic effect. Whereas if it inhibits storage, inhibits the synthesis or blocks storage, it can have an antagonistic effect. Drugs that affect the release of neurotransmitters can have agonistic or antagonistic effects as well. Increasing release, which we'll look at, can cause agonistic effects. Inhibiting release can cause antagonistic effects. And finally, drugs that affect reuptake or the breakdown of the neurotransmitter can have agonist or antagonistic effects, as shown here. This is further detailed in this slide, which shows a more comprehensive look at agonistic drug effects and antagonistic drug effects. So on the agonistic side, we see drugs that increase the synthesis of the neurotransmitter, drugs that increase the number of neurotransmitter molecules by destroying the degrading enzymes, drugs that increase the release of neurotransmitter molecules from the terminal baton, drugs that bind to the autoreceptor and block their inhibitory effect on neurotransmitter release can have an agonistic effect. Drugs can bind to the postsynaptic receptor to activate them, or drugs can block the deactivation of neurotransmitter molecules by blocking degradation or reuptake. I'm not going to go over that for antagonists. You can do that yourself, but I think you get the idea. So let's begin with acetylcholine, which was one of the first elucidated neurotransmitters. Production of acetylcholine occurs in the nerve terminal in the synaptosomal cytoplasm, and it is produced from choline and acetyl-CoA to acetylcholine via the enzyme choline acetyltransferase. This enzymatic transformation is the rate limiting step in production, important to know, and the rate limiting factor is the choline supply. Fortunately, choline can be reused. Now, it is released in a voltage gated calcium dependent fashion, where depolarization results in calcium influx. The synaptic vesicle fuses and releases the acetylcholine. Degradation in the synaptic cleft occurs by acetylcholinesterase. And this slide just demonstrates the 6 steps in that process. Excuse me. So we have the synthesis of acetylcholine, as I pointed out in the last slide. I'd like to note that you can block the transport of choline with hemicolinium. So that would have, right, an antagonistic effect. The uptake is then, the acetylcholine is then put into vesicles, and those vesicles then can be released, and release can be blocked by botulinum toxin, which would have an antagonistic effect, or spider venom can cause a massive release of acetylcholine, which can have an agonistic effect. The acetylcholine then binds to the post-synaptic receptors. There are 2 channels that are involved, ligand channels, muscarinic and nicotinic. We'll look at those. And then degradation of the acetylcholine by acetylcholinesterase occurs in the synaptic cleft, and the choline is subsequently taken up via transporter back into the terminus. So as I mentioned, there are 2 types of receptors, nicotinic and muscarinic. Commonly, these 2 types are found in all preganglionic synapses, both sympathetic and parasympathetic. They are found in the CNS, with the muscarinic receptors predominating over nicotinic. Muscarinic receptors are more exclusively seen in all postganglionic parasympathetic fibers, as well as postganglionic sympathetic fibers at the sweat glands. Nicotinic receptors are seen at the neuromuscular junction, adrenal medulla, and preganglionic fibers, which are exclusively nicotinic. There are several disease states that involve or affect acetylcholine neurotransmission. These can be divided into 2 broad groups: those which cause acetylcholine deficiency, and in the next slide, those which cause acetylcholine excess. So in terms of deficiency, this can happen in a number of different locations. We look at deficiency at the presynaptic neuromuscular junction, blocking release. This is caused by botulinum toxin, Lambert-Eaton antibodies, which block the presynaptic calcium channels, tick paralysis, and sea snake toxin. Postsynaptic blockade of the neuromuscular junction receptor includes myasthenia gravis, depolarizing blockade, such as with succinylcholine, and non-depolarizing blockade, such as occurs with curare. In addition, acetylcholine deficiency is seen in Alzheimer's disease due to degradation of the acetylcholine nuclei. And then in the next slide, we basically see those diseases associated with acetylcholine excess. And these typically involve exposure to either toxin or drugs. So we can have acetylcholinesterase inhibitors. These are drugs or toxins that block or reduce the breakdown. And there's some really classical stories out there that you could read about, including organophosphates, sarin toxicity, black widow spider venom, as well as donepezil toxicity and pyridostigmine toxicity. There's some other special features that you should know about, and that is that the muscarinic receptors are also located in the cardiac conduction system, smooth muscle, and exocrine glands. I've put in this slide, which is 2 mnemonics to remember the muscarinic peripheral effects of cholinesterase inhibitors, right? So you're inhibiting the cholinesterase and increasing the muscarinic peripheral effects. And the 2 mnemonics are sludge for salivation, lacrimation, urination, defecation, GI pain, and